just get a reminder from our team on the mics here. Okay, everything okay? Okay, cool. So with that, I'll introduce Jason. Dr. Jason Pither is the principal investigator in the Biodiversity and Landscape Ecology Research Facility at the Okanagan campus of UBC. His lab engages in both basic and applied ecology research, including biogeography, community ecology, and invasion biology. He serves as an associate editor for Global Ecology and Biogeography and Ecography, and as an advisor to New Phytologist. He is an open science ambassador for the Centre for Open Science and instructs graduate students across Canada on best practices in reproducible research. Please join me in welcoming Dr. Pither as he speaks to assessing transparency and reproducibility in invasion science. Okay, wait. Okay, yeah, you know the system better than me.
Okay. Okay, there we go. And I guess, do I have to use this thing? Or do you have a lapel microphone?
That's being used for the questions. This should work.
Okay. Hello, there we go. Okay, thanks a lot for this opportunity to speak to you today. So yeah, I'm here at this campus, and my students and I have had the privilege of collaborating with Indigenous communities, the province, and the federal government on invasive species research over the last 15 years. And during that time, though, I've had my own journey discovering about open science, and especially in the last 10 years, have developed some expertise in it, and enough to become an instructor in it. And so what I want to talk to you about today is really that process of learning about all that stuff and then sharing what I've learned about it and how it's relevant in particular for invasive species research. And so what this is here is just an idealized sort of pathway towards success in any kind of management context, and you can apply this in invasion research as well. And so ideally, we have stakeholders at every stage of the process inputting into co-developing management strategies, and then designing primary research that tests those strategies. And then in a lot of contexts, it's beneficial to have, build up a body of literature and research that can then be synthesized and inform a sort of synthesis and policy ultimately, which then gets put into practice in terms of management practices. And there would be some adaptive adaptation in there too if we need it, and on a regular basis it gets updated. So that's idealized, and in many, this is applied in many contexts you can imagine. And under the hood, of course, are the details underlying the primary research. And what we've got here on the right-hand side is sort of an idealized research workflow, right? Where we're co-developing those ideas for strategies. We're designing studies, and experiments or exploratory. We're collecting the data and eventually disseminating the data. And over the last 20 years in particular, meta-scientists, those are researchers who research about research and how it's done and how it's disseminated, have discovered that by and large, in many disciplines, the majority of those research outputs are in fact unreliable for a variety of reasons. And a lot of this started in the psychology realm. They were at the forefront of doing real critical assessments of their own body of research and discovering that it was not reliable. And then, of course, in the medical sciences, there are actually what I would consider sort of tragic examples of hundreds of millions of dollars spent on research that is ultimately unreliable. That could include in Alzheimer's research, in cancer research, you name it. And so this is obviously not ideal. So what do we mean by reliable research? Hopefully you can see that text. I'll read it out for you. So first of all, that it's transparent. So that's all aspects of the research, including the design and analysis of decisions and the research outputs, including non-significant findings, are reported in complete, honest, and unbiased way. And then, of course, that the stats and the design of the experiment are themselves sound, and that you have openly provided all the research outputs. So that's non-sensitive data. Of course, there's data that shouldn't be made open. Code and other outputs relevant to the research should all be made open. and that it's reproducible. You've probably heard that word thrown around a lot. In this context, we're talking about independent researchers being able to specifically follow your study design and the protocols using the data that you produced, right? So imagine someone in your lab coming into your lab 2 years later, a new graduate student, and just trying to figure out what you did and being able to do it all, okay? There's a more rigorous form, which is replicability, and that's independent researchers actually doing everything you did on their own and generating their own new data, okay? That's the ideal, but what we're mostly focusing on is just up to that reproducibility level. Okay, so that's some context for you. But the problem is that our current research ecosystem within which we work really, instead of promoting transparent and rigorous and reproducible research, it engenders or even incentivizes workflows in which so-called questionable research practices flourish, and ultimately yielding all this unreliable research. And it can also perpetuate inequities and injustices and biases as well. So that's a clear problem, right? We're all embedded in this research ecosystem where, for example, especially early career researchers are really pushed to publish as much as possible. And publishing takes time, and it especially takes time if you're doing it in a very rigorous, open, and transparent way. And so instead of taking that time, we're encouraged to really just rush things and publish what ultimately is less reliable than we'd like, all right? And I want to emphasize here that, of course, researchers are doing what they think is right because they've learned in a system that sets up these traditional methods. And so it's certainly not on purpose, okay, most of the time. All right, so this is a slide that in my teachings of open science, mostly to graduate students, clearly can be discouraging. And what it's showing is a whole suite of these so-called questionable research practices that can inject themselves into the research workflow and undermine the reliability of research. And you've probably recognized some of these terms. There's terms like, let me see here we are. Statistical power, p-hacking. So p-hacking is when, you're essentially either consciously or subconsciously seeking out significant results in your stats. Hypothesizing after the results are known. So that's, doing all your work and then you find some finding and you sort of subconsciously or consciously say, oh yeah, that was expected all along and I'll present it as though I expected it after the fact, right? So all of these things can really clearly chip away at the reliability of the research outputs. What was really enlightening was a survey study done about 7 or 8 years ago now by some meta-scientists of ecology and evolutionary researchers on these types of questionable research practices. And the upshot was, so remember that psychology was at the forefront of all of this meta-science research. And so the question was, were EEB researchers as vulnerable to these practices as in other disciplines? And it turns out, yes. So for example, they asked whether at any point in their early career, had they reported response variables, not reporting response variables that failed to reach significance, right? So when you use hypothesis testing or when you're using frequentist statistics, that p less than 0.05 approach, the only way that that is a meaningful number is if you're reporting all of the statistics that you're doing, all of the tests. If you do not report all of that, then you cannot interpret reliably those statistical outcomes. And so more than 60% of researchers said that they failed to report all of those results. Another example is rounding off a p-value to meet that magic 0.05 value, right? So these are just examples. It happens. And remember that this isn't necessarily on purpose. This is what we've been trained in a lot of ways to do within the culture of our own labs, for example. And then there's cognitive biases and biases that come about from, for example, low statistical power. And so these are a couple of examples of studies. So I forgot to mention that the QR code that was on the slide, and all of these slides should be available to everybody. I've made them available. Has a library of all of these citations for you to refer to. These are example studies that looked at bias, and the one on your left talks a little bit about exaggeration bias that arises from low statistical power in the studies, primary research studies. The one on the right was co-authored by a colleague here, Melanie Jones, regarding the wood wide web, the mycorrhizal connections in forests by mycorrhizal fungi below ground. And it was an incredible study that really revealed, number one, that a close examination of the research that went into generating this narrative around trees speaking to each other below ground was not at all reliable, mostly because of weaknesses in experimental design. But most enlighteningly, it was the positive citation bias that really exacerbated this narrative. So this, the confirmation bias that goes into favoring outcomes that are in line with what you want to see at the expense of overlooking those that contradict that. All right. So in the end, what we end up with is a literature that is itself demonstrably biased and unreliable. And that sucks as a researcher ourselves, especially as graduate students that want to get guidance and reliable guidance from that literature. And all of that generates what some people are calling research waste. And so the study on the left is just one that I recommend you guys read because it sort of quantifies it in a context of conservation biology, which has a lot of parallels with invasion science. But on the right was a recent study that actually tried to quantify that amount of research waste in ecology and evolution. And shockingly, they discovered that only 11 to 18% of research reaches its full informative value. And that's an important phrase because especially in applied realms like invasion science, informative value means it's usable and reliable, right? So that's pretty depressing. So the upshot is that in many management contexts or intervention contexts, be it medicine, invasion science, you name it, the research that informs synthesis and policy is the majority of it is unreliable. Now, let me emphasize that, of course, that means that there is some that is very reliable. And we can all, I think, point to a lot of research that really stands out. So I'm not diminishing that at all. But the vast majority of the volume of that research is not. So then the question is, well, okay, that's all general EEB research and medicine and all that stuff, but does that actually happen in invasion science? And so we sought to at least get a preliminary idea of that just because Dr. Fabio Milène, who was a former postdoc of mine, who saw his talk maybe the other day, he was conducting a systematic map of research on impacts of invasive plant species in riparian and foreshore ecosystems in BC. And we thought it would just be an easy little side project to just do a quick and dirty assessment of practices in the studies that he was already systematically sort of assessing for these other features. And so this is a little discussion paper that's in revision at Neobiota right now. And in all, he looked at 45 studies on reed canary grass and Russian olive. And remember, this was all part of that other systematic review or systematic map. And evaluated 14 practices that are pretty easy to assess at face value, because this wasn't an in-depth assessment, just to see how we're doing in invasion science. And the upshot is that not particularly well, consistent with every other discipline so far that's been assessed. So on your left here is just the number of publication bar chart over the years. So a handful of studies each year, dating back to 2000 up to 2020 2020. And the black line is the score overall. And on the right-hand side are a list of some of the practices that we're looking at. And so, yeah, not particularly well. And I want to be totally upfront that one of my lab studies was in here from 2015, and we only scored 25%, right? So this is stuff that we're all learning how to do better. And certainly back then, I was nowhere near as aware of a lot of these issues. So I want to emphasize that we're still learning too. You know, it looks like things are doing okay with respect to some of the statistics reporting, saying that they tested assumptions of statistics and so on, but we did not have the means to go into depth in a lot of these things. And we wouldn't be able to because very few papers provided data as well, okay? So the upshot in terms of invasion science is that it looks like from this preliminary studies, it's consistent with what's happening in other disciplines. And we were, of course, assuming that these focused studies on reed canary grass and Russian olive were representative more broadly. Maybe that's not the case, but that's what we assumed. Okay. So for the rest of the talk, I just want to, how am I doing for time? Okay. Just want to quickly, I'm leaving this last point for discussion, but these first two things, how do we make progress then towards this? And I emphasize incrementally, because when you look at this as this whole huge issue that we've got to tackle, it can be a bit daunting. So you just chip away at it. And I'll talk about some of the things we can do, and also tools to and resources to assist with that evidence synthesis, including reliability checks. We need to move toward that in invasion science. All right, so what I'm doing now is just providing some resources to consult with. And the first one is on statistical reporting, practice and reporting. And these are a handful of studies that are really good at providing that kind of guidance. And some of them are a little bit more introductory than others. So that's it? Okay, all right. I'll zip through it. Okay. Okay, yeah, I started early, but that's okay. Okay, all right. This paper in particular is an eye-opener. I recommend everybody read it. I'm still absorbing it, even after 2 years. It emphasizes things like multiple testing being a problem. And that is a rampant problem in invasion science, okay? Data archiving. So even if you're not doing everything great in your study design or in your statistical analyses and your reporting, if you make your data open, it means that meta-scientists or meta-researchers can do meta-analyses down the road using your data. So that's important. And then pre-registration, if you're doing hypothesis testing, which a lot of invasion scientists do, pre-registration is a way to avoid all those biases, documenting what you want to do beforehand, registering it, and then going and doing your research, and then reporting any deviations from your original plans in a transparent way. That's a crucial way to avoid bias in your hypothesis testing research. These are a couple of examples of tools for reporting about research or synthesizing research, but it really emphasizes a lot of the hypothesis testing realm. And the problem is we can't assess the reliability of any of these primary research papers that go into this synthesis. So really cool steps towards this. It would be awesome if we could then come up with a rigor analysis of the underlying data. And then that's what these folks in conservation have been doing. Bill Sutherland and others in Europe, they put together this database that actually leverages all the data from research and is able to do its own rigorous analysis from that primary research. That's what we should move towards. All right, so lastly, here's some resources that we offer at UBC to learn about these things. So there's a QR code there, and again, the slides are all available. And then I've put together a sort of outline of a reproducible research workflow. If you're interested, it gives you all the tools and tips to do that as well. And I just want to emphasize that we're still learning this in my lab. We have not achieved the ideal workflow yet. We haven't done a full pre-registration with a publication, although we have some pre-registrations done. And so it is a journey, and it should be incremental. Just chip away at the things that you can. All right, that's it.
Amazing presentation. Thank you so much, Dr. Pither, not only for sharing those insights, but also some really great tools to help build our knowledge in this area. Any questions from the audience for Jason?
Yeah, okay, there you go. Right at the beginning or this one? Which one?
The very beginning.
So the title slide, yeah. That's it's a terrible library. Should be on
I think it was at the end, right?
We're just looking for.
There you can.
Oh,
Oh.
it's not showing up. Oh, maybe we, so I sent an updated version with this QR code. No, it's not that one. Oh, well, sorry, I sent a version of this slide yesterday that had the QR code, but maybe it didn't get up there. It's
I can pull it up just a
That's
second.
okay. So,
No, you're on.
yeah.
Thanks, Jason. That's a really interesting talk. And I know you're just scratching the surface this morning of everything you've done. I like the idea of sort of the bottom-up approach of training and teaching these best practices. What do you see from sort of the top-down end? Because so much of it happens in terms of what journals ask for and what NSERC requires us to do. What are your thoughts?
there is more to speak. Yeah, so some of the biggest steps recently have been around data management from Tri-Council. And so at least folks are having to submit data management plans, which is an important first step. But there's much left to do. The other key pieces, and the medical researchers in particular, have been doing critical assessments of promotion and tenure practices, and unsurprisingly revealed that there's very little mention of this stuff within promotion and tenure, and that has to change. And there are slow movements towards that, especially in Europe. Unfortunately, we're always lagging behind. So that's another area, but at least the assessment of all of those practices has now been done, and the data are there laid bare for administration to see that this is a critical spot. And so that's where I see the really important ones, because it's only once it's valued for career progression, that we're going to get there. Oh, there we go. Okay, there we go. And journals, yeah. So American Naturalist now has a data editor, which is a big step. More and more journals are now requiring data deposits, but they don't have the capacity to check the usability of the data that is deposited. But it is something that's on minds, and I'm actually just submitting a discussion paper to the new phytologist around this specifically, because I'm an advisor there, and it's really frustrating to not have that. Other questions?
Hi, this is Becca Nelson from Utah State. Great talk, like really cool resources for data reproducibility. I'm really curious if you think the invasion science literature as a whole, like whether the preconceptions around applications to being concerned about like the negative impacts of like the worst of the impacts of introduced species has sort of led to any sort of things similar to with the mycorrhizal example, where maybe more negative or positive effects have been underreported in the literature. Thanks.
Absolutely. Great question. So just like in any discipline that sort of builds up its own theory tenacity, they call it, right? There's lots of that. And that's why, especially, so the folks that put together that really awesome synthesis tool around hypotheses in invasion science, as soon as you have these core hypotheses that have lived in the literature for a long time, you do get these camps that build up attitudes around that. And so it's so crucial to do pre-registration when you're testing those hypotheses with confirmatory research, because otherwise you will succumb to confirmation bias inevitably, even if we don't intend to, right? These are subconscious biases most of the time. And so that's a great point, and invasion science is not immune to those very things that mycorrhizal ecologists have discovered recently too. Yeah.
Hi, I'm Lim from Simon Fraser University. Yeah, super amazing work. I'm really curious. I'm a grad student and reading a lot of papers. Obviously, we do like lab meeting discussions, and often when we look really closely, we find some what kind of feel like fatal flaws often. Not all the time, but I was curious for you as a prof, when you're reading literature, what is your workflow in terms of knowing that most literature out there is maybe unreliable? Are you just doing it super thoroughly and like going through their GitHub and rewriting their code? Are there certain things that you can kind of look for that it'll tip you off if it's a better paper or not? Or like, yeah, I'm just curious, in your capacity as a professor, a busy person, how do you sort out the weeds from the good?
Yeah, great question. I wish I had the capacity to, because I love delving into data. I'm sure a lot of us do here. But there are telltale signs and flags. And so if if it's a research topic that is absolutely core to what we happen to be working on, I'll take the time to do that and hope that the data and code are available. If I see issues, I can get into it. But most of the time they're not. And so you sort of stack up a pile of papers that you don't discard. There's value in all research, right? But you have to just be skeptical, and you lay it against the minority of papers in which all the practices are reliable. And it's a great learning moment for all graduate students, right? When you can compare and contrast studies that are tackling the same question or hypothesis and how they've gone about it differently. So the short answer is, I wish I had time to do more of that deep investigative work. Most of the time, what I do is I say, okay, for example, if it's clear that they haven't used the right statistical models for the data at hand, they probably haven't received the training that ideally they would have, and therefore everything downstream from that is a little less reliable for me. Again, you don't throw it out, but you read it through that lens, right? So that's a great question, and that's how I sort of try and balance it, because otherwise it can get pretty depressing if you just throw everything out. Yeah. Okay, great, thank you. There's the QR code for the Zotero library. It's a huge library of open science stuff, but these papers were in there, so you just search by name.
All right, thank you so much, Jason. All right, let's try that again. Our next presenter is Brennan Webster, who's been with the BC Ministry of Forests Invasive Plant Program since 2019. Brennan is the program's invasive species data specialist, where he combines his invasive plant management knowledge, GIS, and data development skill sets. Brennan has been involved with the development of InvasivesBC for the past couple of years and was a key member of the team leading the transition from the former Invasive Alien Plant Program, or the IAP database, over to the new system, both on the development side and supporting users. Please join me in welcoming Brennan, who is going to be sharing more about his work online.
Thanks. Here, I'm going to share my screen. Hopefully that's coming through there.
Yes, we can see your screen and we can hear you. You're good to go.
Awesome, thank you. Yeah, hi everyone. I'm Brennan Webster. I'm the invasive species data specialist for the Ministry of Forests Invasive Plant Program. My goal for this talk is basically to bring awareness to the large invasive plant data set our program has accumulated over the years. There's a lot of sort of operational invasive plant work, surveys, treatments going on every year, on the order of tens of thousands of records being entered into the database. Next slide. So first, I'm going to talk a bit about the user group for IAP and InvasivesBC. I'm going to talk about IAP, which was the previous database that we recently decommissioned. I'm going to talk about the new database, what's different with it, specifically the transition from point data to polygon data. I'm going to talk about the structure of the data a bit, Some of the new components of the system that are different from the previous database, and also talk about ways that people can extract the data from the database. The BCGW layer, if anybody's familiar with the BCGW or the BC Data Catalog, it's publicly available provincial data sets. And then at the end, hopefully we'll have time for some questions. So the user group, I'll talk about this just so people get a sense of who's using the database, what they're using it for. It's kind of the main database in the province for people who are doing invasive plant work. Most of that work will end up in this database. So the regional invasive species organizations is one of the main users of the database. They correspond generally to regional districts. So a regional district generally will have a regional invasive species organization area, and they'll generate priority site lists at the start of the field season based on their area, based on the species and the jurisdiction, and all of their data for the field season will end up getting uploaded into the database. In some cases, some regional districts don't have a regional invasive species organization, and the regional district will be doing that work themselves. There's also all of our program staff, summer students that do field work on priority species, also enter all that data into the database. Municipal governments as well are sort of similar to the regional districts. And then there's also forestry, mining companies, and utilities that have an obligation during their operations to, if they notice invasive species, they have to send the report to us, and they also have an obligation to treat. And then there's also the Report Invasives app that we have that is sort of a citizen science type of thing where the public can send us a report of an invasive species. It's pretty basic. They send us a picture generally and then a location, and our staff will go through it, verify it, and enter it into the database as well. So that data makes it in there too. So the Invasive Alien Plant Program database, most people who know about it refer to it as IAP, was the original IP program database. It started in 2005, and we decommissioned it in 2023. At the time it was decommissioned, it had about 500 users, and the data set included just under 160,000 sites. I'll talk a bit about what the data structure was, what a site actually is. So the location of invasive plants it's sort of tied to a single point location, and any surveys or treatments or monitoring or biocontrol activities are consolidated onto that single location. So there's not a lot of spatial information surrounding that. You can infer based on some of the attributes. So the data was a plant species generally, and then an area that the user entered, as well as like comments and density and distribution. So in order to get the information sort of aggregated on the site, we had what we call the 100-meter rule. So if you're creating a record within 100 meters of a site, that information should be consolidated onto that site to avoid having duplicate sites in the same area. But it was sort of more of a guideline and not a strictly enforced rule. So if in the screenshot here is what the front end of the database looked like. This is where people would fill out records and enter them into the database. If you did it through the front end, like in this screenshot, you would be warned if you were entering data that's within 100 meters of a site, and it would do it for you. It would enter it onto that site. But there's also another method of uploading the data that, like the regional invasive species organization areas and the regional districts, they're generating a lot of data. They don't want to add the records one by one through the front end. So there was a spreadsheet, sort of standardized spreadsheet batch uploader, and the batch uploaded records sort of circumvented the 100-meter rule. So we did end up with a lot of sort of duplicate sites, or sites stacked on top of each other, or clustered more than they should have been. Another issue that we had with IAP was there was no data collection component. That kind of goes back to what I was mentioning before about people batch uploading data. Because the front end of the database is intended for use on an internet browser with an internet connection, there was no functionality for offline record creation or like caching data in the field. So generally, all the user groups would use a third-party data collection app, and that sort of comes along with its own problems. For one, you're using this third-party app and creating all this data, generally trying to keep it in the same format the database accepts, but it still in the end requires a bunch of data cleaning. And also, if you haven't modeled it exactly like the database, you end up with sort of validation errors and stuff like that. Or people after the fact, when they're in the office, they realize they didn't fill out everything they needed to. They're sort of creating data, fudging data after the fact, which comes with some issues as well. IAP also had a separate web map, which if anybody has used IMAP-BC, it was basically like that. It's an Esri-based product. It was sort of limited. You couldn't filter things on the map. You were just shown the whole data set. And there were some sort of pre-cooked views of the data, but it wasn't very customizable. You just basically saw everything on the map at once. And that gets me to the new database, InvasivesBC. So we created this with the intention of sort of solving some of the issues that we had with IAP. One of the main changes is most of the data in InvasivesBC is polygon data. A lot of the structure of the data as far as invasive plants and the attribute fields is the same. The map, this is just a screenshot of what the main page, the records page on InvasivesBC looks like. So the map is integrated with the data. You see at the bottom the record sets, which will just show you a table view of all the data, and you'll see it on the map as well. So you can filter the record sets. It will show you those rows, and it will also show you them on the map. The database is has PostGIS enabled, so there's a lot of functionality for users to use like KMLs or drawn shapes to filter their data. For instance, you can upload like an area of interest KML, and the database will filter it for you. The map is pretty high performance compared to like IMAP-BC. It uses vector tiles sent from the database, and can display like 200,000 points on the map pretty quickly. It's one of the big improvements over the old system. So here I'm just going to give a bit of a demonstration on the difference between the point data and the polygon data. So to make this, I just ran a query of IAP sites from the old database that have been superseded by observations from the new database. So this is a point site that somebody made. And when you're looking at this, you need to understand, I guess, that they also inputted an area, which kind of gives you an idea of how big the site might be. So say this person entered this site and said the area was a hectare. You still can't really understand what the footprint is just based on the point itself. There's a comments field. So for single records, you could read the comments and it would say like, oh, looks like it's in a wetland. Maybe it's along the shore. That's where the infestation is. But when you're trying to do like a big data analysis on all the data, you can't use those sort of open-ended text fields to do that. It's kind of impossible, like keyword searching or stuff like that. So it's better to have the polygon footprint. So the polygon looks like this. I'm sure everybody knew what that was going to look like. And I'll just...
Warning.
Sorry, pardon me?
Sorry to interrupt. This is your 2-minute warning.
2-minute warning? Okay. I'll just go through. There's another point. This is the polygon. This one looks like it might be on a road, but actually it's in this field. That's another issue with the sites is they're stuck in the same location. You survey them every year. The data is getting just stuck to the same point, whereas the infestation could have moved. It gets treated in some spots. Maybe the person didn't do a full extent survey, so it's still growing somewhere else. That's something we see a lot. So landfill. So that one was kind of obvious. This one, a road again, but it's actually in the field. Sort of a road there. That makes sense. Yeah, so you can kind of see that there's just a whole nother level of detail when you have the actual footprint of the infestation instead of just a point that everything is attached to. Yeah, people can draw some insane shapes as well. This is one of my favorites. So yeah, like I said before, the data specifications are very similar. One change we made is you can make positive or negative observations. Positive just means you found the plant. Negative means you didn't. Another big add-on to this was the mobile app, which has offline record creation and the ability to cache records and cache base maps so you can use it in the field. So a lot of the work that we do is done with spotty service or no service, so it's kind of a requirement. And that's sort of the final piece of the system where everything is within the system. Nobody has to use a third-party app like Avenza Maps or Field Maps or something like that. And you can also generate reports. All of them at this moment are CSV reports. They actually come with the geometry in a field. It's a well-known text format, which is just a text representation of a geometry. So you can sort of easily convert those from the spreadsheet into something like a shapefile or KML. And then finally, the other way to get the data is in the BCGW. All the IAP data is in there, and also all the InvasivesBC observation data is in the BCGW, or the data catalog. I think they're sort of interchangeable. And you can also get it through IMapBC as well, which is what is in the screenshot. And it's updated every night at midnight, I think. And now I'll take questions if anybody has any.
Fantastic. Thank you so much, Brennan. It's great to learn a little bit more about the process behind this new tool. And you did get some good laughs in the room about those insane shapes. What a great slide. We've got a question coming for you here, and we just have time for one or two. But please go ahead.
Well, I'm Adam Soames from TRU. I've used some of this data to find sites for knotweed infestations. And one of the things that I would notice is that you go to a lot of the sites and the infestation has been taken care of. So it's actually gone. I was just wondering what kind of controls you've got to like get rid of information on like sites that might have been treated or those weeds are no longer present.
Yeah, so for invasive species specifically, sort of best practices or the guidance we give to users is if you go to an infestation, you don't find it, you're supposed to make a polygon covering the old polygon and say it's a negative observation. So yeah, it seems like it's possible that you were the first to survey it after it was treated and gotten rid of. But it's also possible that users aren't following that guidance. And so the positive observation is, yeah, the most recent one.
Thanks for that. Some user support in real time. Any other questions for Brendan? Oh, we got one ready to go here.
My name is Mandeep from Soil Tracker Solutions, Inc. I'm just wondering, do you guys work with any other agencies like BC One Call? They also use a polygon system on their map. When, you know, as contractors, you have to draw what you're going to be digging. And would that be helpful for you guys? Because they're kind of using the same system. With, if there was invasive species where they're going to do the dig, would that help you guys?
Yeah, no, I haven't actually heard of them. That's interesting. Yeah, sorry, can you repeat that as BC One Call is what you said?
One Call is like any contract, anybody that has to do any sort of digging, they have to go to BC One Call, make sure there's no utilities or anything else in that area. And then you put polygon map. So you have to draw what area you're going to be excavating and so forth. I think that would be beneficial for you also if you guys could incorporate the invasive species map with that.
Yeah, no, that's definitely interesting. I'll look into it. I'd never heard of that before. Thanks.
Sorry, just Abdul from Soil Trackers again. Just so, like, do you guys offer API like connectivity or any third-party ability to connect to the system to pull that data without doing manual uploads or downloads?
Not at the moment. We don't have an API. There are plans to make one. The best way right now is probably to use the BCGW or the BC Data Catalog. It's publicly available, and I think you can get WMS service from there, and that's probably the closest, like, sort of automated solution.
Great to see some of these idea sharings happening right here. I think this would be a good thing to connect offline on and follow up on. So I'm going to welcome Nick to come up and introduce our next session. But one last round of applause for Brennan. Brennan, thank you so much for joining us and sharing this really, really important information.
All right. Hello again, everyone. I will keep this quick. Yeah, so we're moving into session 7 with a look at some impacts. We're going to hear about pollinators and crabs, which are my personal favorites. No bias there. Sorry, Jason. So our first speaker is Dr. Sandra Gillespie, who's an associate professor in the biology department at the University of Fraser Valley. She completed her PhD at the University of Massachusetts Amherst in organismic and evolutionary biology and has held postdoc positions at both University of California, Davis and Simon Fraser University. Her research examines the role of native pollinators in agriculture and the threats facing wild bees. She gets to work with farmers and other knowledge users to examine the role of native pollinators in pollinator-dependent crops, such as blueberry and cranberry in the Fraser Valley, and has studied pathogens, parasites, pollinator community structure, and the relationships between invasive plants. Yeah, please give a warm welcome to Dr. Gillespie.
All right, I'm going to see which microphone option. And everyone, all right, if I kind of hug the mic at the front here? We all right? I'll resist my urge to pace around aggressively. So thank you all for coming. Oh,
Just a quick adjustment. Okay. It's not getting shared.
I'm going to talk a little bit about a project that's in progress with collaborators in the United States, looking at a pollinator that is not meant to be here. So down is not working. Unless it's very slow. We have to continue the tradition of technical issues through the final day. There it goes.
Just a sec.
It will only work for you. I've had classrooms where the screen was actually upside down. Luckily, my students have a sense of humor, but this isn't so bad. At least nobody's trying to read the screen.
There we go.
Lovely, thank you. All right, so bumblebees are a very important pollinator, both here in North America and elsewhere, particularly in temperate regions. And if you work in the sort of conservation side of things, you know that population declines in bumblebees and in pollinators in general have been broadly documented. However, those declines are not consistent across taxa. And in fact, there are winners, quote unquote, and losers across different species of bumblebees. So for example, on the East Coast at the top there, Bombus affinis is declining and has declined dramatically, while Bombus impatiens has increased. But bumblebees are also an agricultural tool and an invasive species. So in the 1990s, two companies developed methods for commercial rearing of bumblebees. In Europe, Bombus terrestris is the primary bee that's used. And in North America, we have Bombus impatiens and originally also a Western bumblebee species called Bombus occidentalis. And terrestris particularly has been moved globally. It's invasive in Japan, Israel, New Zealand, Tasmania, and South America. And in these places, there are documented impacts on the native species. In Japan, Bombus terrestris not only competes with native species for resources, but it's more alarmingly been found to hybridize with native species. In South America, there's actually 2 European bumblebees that have spread widely. And where they spread, there's only one native bumblebee in South America. It's this beautiful Bombus jalbalmi. And where the introduced species spread, the native species disappears. And in this case, we have some evidence that pathogen spillover may be playing a role. So what is this? Pathogen spillover occurs when managed species spread disease to nearby wild species. And in the case of South America, there's spillover of a pathogen called Apocystis bombi that seems likely to explain the declines of the native species. But we've also documented pathogen spillover from managed bumblebees in North America, seeing high rates of pathogen infection, for example, near greenhouses that are using Bombus impatiens in Ontario. So what is Bombus impatiens? It's a really pretty bee. Most bumblebees are cute, I'm biased. It's native to Eastern North America, and until recently, it was the only commercially available bumblebee for particularly tomato pollination in North America. In BC, it's been used to pollinate greenhouse tomatoes since the loss of commercial Bombus occidentalis. Rearing of Bombus occidentalis collapsed due to an outbreak of pathogens in rearing facilities, and it's never recovered. And so how did we wind up with this eastern bumblebee in huge numbers in greenhouses in BC? Well, there's no regulations regarding the movement of beneficial insects such as native pollinators. I think it reflects or mirrors some of the things we were talking about with biofertilizers on the first day, that we have these tools that seem natural, that are beneficial to agriculture, and we're not sort of digging into the ecology behind what else might be going on. Now, at the time, there were measures agreed upon to minimize risk of escape, including queen exclusions and procedures for disposing of colonies at the end of their use. Despite these measures, it was first documented to have escaped in 2003 in Delta. And since then, the population has become naturalized and has spread in BC and south into Washington. What are the implications? It's just another bee. Bees are nice. Well, to sort of understand this, I'm going to give you a little primer on a bumblebee life cycle. So bumblebees are a lot like an annual plant. We have queens overwintering, and they emerge in the spring to search for nests. Wonky slides, the theme continues, it's fine. Oh, my arrows are all over the place, that's amazing. Anyways, the queen will rear her first brood and establish a colony once she's found a nest site, and then the colony will grow, produce generations of workers, increase in size until it reaches some size threshold where they can produce males and queens. Mating occurs, everybody dies except for the queens, who overwinter and start the cycle again in the following spring. And in the context of this life cycle, we can think about some major ways that different species of bumblebees can interact with one another and ways that a non-native species might impact our natives. One is through resource competition, first for nest sites, which is very hard to study, but likely nest sites are a limiting factor for bee populations. And secondly, for floral resources. Floral resources are very critical for queens when they're first establishing their nests, but they're also important for that process of colony growth later in the season. Secondly, of course, you could have pathogen spillover. And various pathogens in bumblebees are very well studied and have documented impacts throughout the life cycle, doing things like reducing worker lifespan, reducing the success of overwintering of queens, or reducing the ability of queens to establish new nests in the spring. So I'm going to tell you just a little bit about what we know about what's going on right now. So what is Bombus impatiens even doing in BC? I'm going to talk a little bit about geographic spread, both current and potential, and what information we have about impacts on native bees. Do we have evidence for competition for resources? And can we tell if Bombus impatiens is a source of pathogen infection? Okay. So 2003, I have a laser pointer. Yeah, there's the first documented point. All this data is from iNaturalist. 2003 to 2015, you have a few more observations across the lower mainland. 2016, a few more. And then I'm going to jump ahead in time to 2020, when you have sort of documented spread south and a little jump, a few spots on Vancouver Island as well. And this is the current map from 2024. And so the species has spread, but I'd also like to note that public awareness of bumblebees and the data that's being collected on bumblebees is also increased. So we have to sort of remember that people are also aware that we have an invasive bumblebee or are more interested in bumblebees in general. So iNaturalist records of bumblebees in general have also increased. But we do know that it's spread and that it's spread to the south. My American colleagues are not pleased, and we're trying to figure out what's going to happen. So how far can it go? So this is data from habitat suitability modeling that my colleagues did. It took data based on the distribution of Bombus impatiens to the east. And notice, it's quite a widespread bee, which is, you don't want a nice habitat generalist for your invasive, but that's what we've got. So it took data on the known distribution and used it to predict where it could occur in BC. And if we zoom in on our predictions, the invasive range could expand considerably. There's lots of suitable habitat in Washington and Oregon and down into Northern California. And then in British Columbia, throughout the Fraser Valley and Vancouver Island as well, and perhaps up the coast, though it's, some of this, it's a little bit hard to know from these models. But clearly, the potential range of this species is much greater than its current range. It's likely to continue spreading. Okay. So what do we know about impacts on native bees? This is something that myself and my colleagues and other researchers at UBC are only beginning to study. So there's a lot of sort of preliminary starting data here. So first, let's think about competition for resources. And what does that look like? So we might expect a bumblebee has many different things to forage on. You've already seen networks this week, which is very nice. So a native bombus might love to forage on a really high quality resource, and it might visit these lower quality resources in lower frequency. When you add a competitor, it's forced to shift because the competitor is depleting that high quality resource and forcing the native bombus to shift to lower quality food. So you might expect a shift in sort of food niches and food breadth in the presence of the invasive. And I messaged my collaborator who's doing this analysis, and she says, well, this is what I can tell you. We have a preliminary analysis that suggests that native bombus may be shifting resource use in response to increasing bombus impatiens abundance. So some support using network analyses for this pattern. Now, I will say that that data is collected in Delta. And in Delta, we have two things going on. We have naturalized populations, and then we have greenhouses full of bombus impatiens, and the workers are flying out of the greenhouses and foraging. So you have high concentrations of these bees from two sources. But as we were doing surveys elsewhere on the landscape, we had trouble finding it, which is interesting when you're looking for a very bad invasive and you can't find it. So what else is going on? And to answer that question, I can go to this really excellent data collected by Melissa Platsko, who's a master's student in Risa Sargent's lab. And she looked at phenology and resource use of different species across the summer and found that impatiens in pink peaked significantly later during the season than other bumblebees. Oops. So just for context, that is September 7th, Julian date 250. So it's overlapping mostly with native species as Bombus vosnesenskyii, and many of these other native species are kind of done with their life cycle before Bombus impatiens becomes really common. So that's interesting. And this aligns with other observations of Bombus impatiens foraging into September or even October. In my parents' garden on the raspberry plants that are blooming in October, and you've got male Bombus impatiens just hanging out. There may be some phenological differences between the invasive and some of our native species that might mitigate competition. What about pathogens? So I've been monitoring bumblebees in the Fraser Valley for the last 7 years because why not? And my data suggests that Bombus impatiens here has higher infection. Oh boy, these are totally wonky. Higher infection of one of the pathogens I was studying, Crothidia, as compared to other species. I've also noticed that infection by this pathogen has increased since 2018. And that coincides with the spread of Bombus impatiens, but it coincides with a lot of other things that are happening. And data collected by another student, I love that I have so many other people to talk about who are starting to study this. UBC Jenna showed that impatiens in her data set actually had lower infection rates overall. Now, her data, again, was sampled in a single year in Delta and included a mix of greenhouse escaped and naturalized bees. So that distinction between these sort of greenhouse escaped bees that are living in boxes and the ones that are naturalized colonies, I think is an important one to start to consider. Okay, so what does it all mean? Maybe there's some resource competition happening between foraging workers, right? So Bombus impatiens may cause native bees to shift resource use when they're present in high numbers. However, phenological differences may be mitigating the impact on some of our native species that are foraging earlier in the season. Pathogen spillover, we have mixed evidence of the potential for pathogen spillover, and I do think more work is needed on this to differentiate between feral colonies versus managed colonies and sort of some of the distinctions of what's going on there. And I'd also like to point out that most of this research is focused on this stage in bumblebee life cycles because it's the easiest time to study. You can go out, you can look at what the workers are doing. So we can go into the field, we say workers are this infected, they're foraging on these plants. But impacts could be occurring at other sort of very key choke points in the life cycle that are really a lot more difficult to study. Okay, so we have many, many questions still to answer. We're doing some work to try to understand where on the landscape Bombus is most prevalent. And whether there's this association with late season urban suburban gardens, there seems to be an association with invasive plants that flower later in the season. And trying to understand whether this phenological shift does decrease the risk of impacts on native bees. So things like competition for nest sites are something that we should start to consider. Furthermore, differentiating between feral colonies versus greenhouse escaped bees should give us a more nuanced understanding of what's going to happen as this species spreads farther south or onto Vancouver Island. And digging into these nuances of pathogen infection are also really important. And I think in general, expanding outside of this sort of easy to study window of colony growth and worker production to thinking about what's happening with the dynamics of queens in early spring would be really valuable. Okay, so just to wrap up, my work, I have 7 years of monitoring data, which means 7 years of really excellent undergrads who have supported this work. These are my collaborators on the big grant that is studying Bombus impatiens. We have a bunch of other work going on in population genetics that I didn't have time to talk about. Yeah, and I'm happy to take any questions. Thank you.
Really fascinating talk. Thank you. I'm super intrigued by your comment at the end about looking into landscape prevalence. And I'm also curious if you think the degree of fragmentation in the landscape, as well as sort of the patchiness of different land use types, might further affect the amount of disease spillover between the bumblebees. Thank you.
Yeah, there's definitely, so one of the things about pathogens is they're pretty well studied from a conservation perspective. And landscape almost certainly has an effect. Pathogens are also affected by stress. Some species more infected than others. Infection changes over time. And the dynamics seem to be really different in different places. So the patterns I see here are really different from what I saw in Massachusetts and are different from what maybe surveys in central US have seen. So there's a lot of question marks. I think the landscape, it's something we're working on for sure. So thinking about land use fragmentation, urban or suburban habitats might support the invasive with that late phenology when most native plants are no longer flowering. And that's sort of another question that's, yeah, we're working on that one for sure.
Hello?
Thanks.
Okay. Hi, my name is Max. I really love this talk. This is really cool. And I was wondering, in a time when pollinators are declining so fast, and these pollinators are peaking at a different time than a lot of the native species, I was wondering if there are some positive impacts of the species, or are they just supporting the invasive plant species that are flowering later?
That's a really great question. I think Melissa's thesis probably answers that better than I could. So I would suggest looking for her papers because she did a really good in-depth look of networks and resource use later into the season. When I talk to farmers, they say, well, does it pollinate? And I go, well, yes. And they go, but I think it's important to remember that though it is not overlapping in terms of the peak of the abundance of workers with some of our native species, that doesn't mean it's not impacting them. So things like pathogen transmission happen at flowers, and there's a potential for interference with things like colony establishment that will affect the abundance of pollinators earlier in the season, despite the fact that we're not actually overlapping with the peak abundance of impatiens. So some of that nuance I think is really important to dig into before we sort of, because yeah, for my parents, they're like, I'm not pollinating our raspberries. It's nice. There weren't any bees late in the year because even Vosnesenskii is done by September, right? So it's complicated. It's kind of a theme. Yeah. You're welcome. All right. Thank you.
All right, thank you so much, Sandra, and the questions there. All righty, next we have M. Lim, who is a PhD candidate from Simon Fraser University under the supervision of Dr. Cote. M is an avid scuba diver and naturalist who is passionate about marine conservation. Their undergraduate research focused on green crabs and mud snails, and now has extended their graduate work on animal-mediated nutrient cycling to consider the role of invasive marine species play in disrupting or bolstering these cycles. So please give a warm welcome to M and take her away.
Great, thank you so much. I will also try to keep myself glued to this general area and resist my also tendency to roam around a little bit. So thanks all for sticking it out to the last day of the conference. I'm pretty excited to talk about my PhD research with all of you. So the invasive green crab is an invader so notorious, I would bet money that everyone in this room and online has heard about them at least in the last 24 hours. So as Steph Green mentioned in her wonderful keynote talk yesterday, green crabs have the capacity to consume large quantities of prey. This has made them a real problem for aquaculture, both on the east and west coast of North America. And further, while they forage for these prey species, they're also tearing up eelgrass, which has really negative consequences for all sorts of really important species that rely on eelgrass habitat as their nursery grounds, including commercially important species like salmon and Dungeness crabs. And across their invaded range, these really horrible invaders are also pretty great at outcompeting native species, which overall makes them pretty undesirable. And again, as I've said, they're on that top 100 list of worst invaders. However, as green crabs consume and assimilate nutrients, they're also excreting metabolic waste in the form of ammonium. And ammonium is an excellent form of fertilizer and is actually the most preferred type of nitrogen for producers to uptake. And so this completes the cycle termed consumer-mediated nutrient cycling. And I wondered if this might actually be a potential positive effect of these otherwise pretty horrible crabs. So that led me to ask this question out on Vancouver Island in Barkley Sound, where I've been based on the traditional territories of the Hawaiian First Nation. And I've been curious whether these green crabs might be able to alter natural nutrient cycling by native species. So Barkley Sound's really neat because there's some areas that have really high populations of green crabs, but there's still some regions that haven't maybe been invaded or have very low populations of green crabs. And so I'm comparing these invaders to 3 species of native crabs, are graceful rock crabs, Dungeness crabs, and red rock crabs. And so I was really curious whether these green crabs might pee more than native crab species, both on an individual level and also among communities. So at the individual scale, I hypothesized that native crab species will probably be peeing more per crab because our native crabs are a lot larger and how much you pee correlates really strongly with how big you are. However, among communities, I thought that green crabs might actually be able to contribute more nitrogen to an ecosystem, just given their really high abundances. So first, I'll be talking about my first question. So we went out and went scuba diving and crab trapping to collect 20 individuals of each species. We then measured these crabs and weighed them before putting them into plastic Ziploc bags filled with filtered seawater. And we took seawater samples at the beginning and end of a 30-minute incubation, and then used fluorometric techniques in the lab so we could measure how much ammonium was in the water before and after the incubation, which allowed us to calculate how much each of these crabs was peeing. And then at the end, we dried and ashed the crabs in order to calculate the dry tissue mass. If you want to find me later, I'll tell you about how this led to an evacuation of our entire department. But I'll show you some results now. So here, our first figure on the x-axis, we can see the log transformed dry tissue mass. And as that dry tissue mass increases, we see an increase in the log excretion rate measured in micromoles of ammonium per hour. And so for our 3 native crab species, shown here with our purple, red, and tan lines, we can see that these native crab species have a higher excretion rate than our invasive green crab shown here in green. So that is really, really interesting because it means that, oh, down is forward. It means that if you were to consider two crabs of the exact same size, our native species will be peeing more than the invasive species because of this elevated excretion rate. But if you imagine a red rock crab, this is not an arrow. This is not an error in the slide. This is how big they are in relation to a smaller green crab. These larger native species are going to be peeing a lot more than the invasive green crabs at the larger end of their size ranges. So to answer the question, are green crabs peeing more than native species? No, they are peeing less per gram, and they're also peeing less per crab. But it still remains to be seen whether that might scale up to a different answer at our community scale. So I thought I was going to have to go out and catch a bunch of crabs and measure them and collect all my own community data. But kind of in the nick of time, a wonderful scientist named Dr. Brett Howard pointed me towards this amazing data set from the Department of Fisheries and Oceans that spans 16 years and the entire coast of British Columbia. I'm just focusing on Barkley Sound because that's where all my crabs were collected from and it's most ecologically relevant. But they've been going out and setting a line of 6 traps and then letting the traps sit for 24 hours before returning to collect the crabs and measure, count, and identify all of them. So very thankful to everyone and all the volunteers from DFO for collecting this amazing data set. Sometimes the traps had upwards of 500 crabs, so that's a lot of measurements. But it meant that I was able to ask some cool community questions without having to measure 500 crabs myself. So for each of the crabs that DFO caught, I can use the species and the size of that crab in combination with my excretion rate model to estimate about how much that crab would be peeing in the environment. And so if you could imagine traps full of crabs, for each individual crab, I can calculate how much that crab was peeing, and then I can add that all up to get an estimate of community level excretion. So now I can ask, are green crabs peeing more? Are green crab communities peeing more? Wait, what is a green crab community? What does that even mean? It should be really simple. We've got green crab communities and we have native crab communities, really obvious. Unfortunately, that's not really how it works out in nature. You see more mixed communities where you might have some native species and some invasive species. You also see communities that have very different levels of abundance. And so it's finding it really challenging to set a single criteria to kind of tell me, is it a green crab community or not? And so that's where I used a pretty common machine learning algorithm called k-means clustering. And k-means clusters are really helpful for taking data and kind of lumping them into groups such that objects in the same group are as similar as possible. So for my purposes, this was helpful in taking green crab communities and putting all of the crab species abundance into this model and asking it, you tell me, what is a green crab community? So my k-means cluster analysis identified 4 kind of distinct clusterings of crab communities. So if you imagine a giant point cloud of lots of different variables, this graph has just taken all of those and smushed them into 2 dimensions. So we can kind of visualize them in human readable space. So the individual points are showing kind of my raw community data clustered and color-coded by the type of community they fall into. So we've got our native high communities, which have higher abundances of mostly native species, and then our native low communities, relatively lower abundance, but still mostly native, followed by lower abundance green crab communities, and finally our kind of classic green crab super abundant community, which can reach these overwhelming densities. So now that I've defined what these kind of communities might look like, I can ask questions about whether they vary in terms of catch rates and also, of course, excretion. So here I'll show you the first figure from that. We've got the cluster on the x-axis, and these colors will be consistent. And then I've got the total catch on the y-axis. So if you were to pull up 6 crab traps after 24 hours, how many crabs are you going to see? So first, we've got our lower end of the communities, so our low native community in light purple, we can see that we were catching fewer numbers of crabs compared to our low green crab community. And then for the higher end of our communities, we've got our native high shown in dark purple, and then that really, really high green point all the way up here is our green high community. As we expected, when there's lots of green crabs, there's lots of green crabs. So this wasn't particularly surprising, but it's most exciting in contrast to our excretion data, where we calculated the total excretion for the community. So looking at our lower abundance communities, we actually saw an opposite trend looking at total excretion. So for the low native community, they might have fewer crabs, but they're actually excreting more ammonium at a community scale than our low invasive community. All right. Oh, too far. Too much data. Come on, come on. Aha. All right, so here's our punchline. After a little bit of delay. Oh, wow, this is crazy. Come on, there we go. So after much suspense, here's our punchline result. So our native high community, despite having relatively way fewer crabs, is producing more ammonium than our high abundance green crab community, seen here. So this points to potentially a trade-off in terms of the number of crabs that we're seeing in communities versus the size and species of these crabs. Whew, that was a lot. Sure, I'm pointing at the wrong angle. Okay, so to dig a bit further into these kind of size versus abundance trade-offs, I plotted the same raw data as an NMDS plot. So again, if you can imagine, big cluster of point clouds, lots of different variables getting smushed into 2 dimensions. These points are again correlated or color-coded by the cluster. And this ordination technique optimizes the distances between points such that points that are closer together are more similar and points that are farther apart are more dissimilar. So on top of this point cloud, I can plot the arrows correlating with how much each species is contributing to the total excretion of a community. And so the direction and magnitude of the arrows correlates with how strongly that species is correlated across the 2 ordination axes. So I'll just point out as a nice logical check that our green crab arrow is pointing towards this big cluster of green crab dominant sites. Now, if we plot that exact same community data, we can now overlay arrows to show how variables like the mean size of crabs in a community and the total catch in a community correlate with these exact same ordination axes. So this mean size arrow is pointing the same direction as our red rock crab arrow, which tells us that communities that received a lot of excretion from red rock crabs in particular tended to have larger crabs on average. And our total catch arrow over here is pointing the same direction as this green crab arrow, which tells us that communities that got a lot of their excretion from green crabs tended to have more crabs on average. And so this just, again, illustrates this trade-off between more abundant crabs and larger crabs. So are green crabs peeing more than native crabs among communities? Also no. And are they peeing more than native crabs in general? Not at all. So this is not great news for native communities. So if you imagined a native healthy ecosystem with native crabs, they're going to be receiving nutrients from the ecosystem by eating lots of prey. However, they're going to be recycling those nutrients in the form of ammonium back into the ecosystem, kind of maintaining a healthy balance. However, if you replace those crabs with many, many, many small green crabs, they're going to be taking actually more nutrients and more resources out of that ecosystem, but they're not returning the nutrients in the form of ammonium at the same rate. So our green crabs are really taking more than they're giving to an ecosystem. And one of the takeaways from this is just the importance of considering not only consumptive, top-down effects of invaders, but also these bottom-up resource provisioning effects, because they might be helping to fill out the larger picture of the impacts these invasive species have at an ecosystem scale. So with that, I'd like to say a big thank you to our funders and to the whole crab team that helped me wrangle crabs all summer. And I would love to answer any of your questions. Thanks.
Hello. Thanks, Em. That was a great talk. Just wondering about the trapping program that you pulled the data in for the second part. I wonder if you could speak to like the design of the traps and the depths that they were deployed at and how likely they are to catch that range of species. I just wonder if you were able to pull in additional trapping data, you might actually see the difference is magnified even more. What are your thoughts?
Yeah, absolutely. So, oh, my pocket slides just disappeared. Oh, shoot. Well, they're gone. So the DFO trapping program is targeting green crab specifically. So the traps are set in the low intertidal to high subtidal. So maybe as the tide goes out, like just barely exposed. And so they are kind of targeting mostly green crab habitat. And I totally agree. Oh, they're back. Magic. So this is the greater map of all those sites where they're distributed. And yeah, I agree. I think if you trapped deeper, you would absolutely catch larger and more abundant native crabs. The one reason that actually kind of worked out for me, so if I'm thinking about ecosystems kind of with and without green crabs, I think those shallower communities kind of are where the green crabs are taking over. And so if I'm thinking about with or without green crabs, that trapping data does address that question very well. But if you're asking about what are native crabs doing in general and overall, then I 100% agree. I think if you got those deeper traps, you'd be pulling up bigger Dungeness crabs in particular. And I think that, yeah, our estimates of native crab nutrient cycling are likely underestimates because they're only considering kind of the shallower ecosystem. Yeah, this data set is incredible, by the way. Everyone should go play with it.
Thank you. Great talk. Adam Soames from TRU. I was wondering, you were talking about green crabs reducing like nutrient inputs into the system due to reduced ammonia. Have you looked at all into nutrient cycling due to like the end of their life, like being consumed by other organisms, stuff like that in the systems?
Yeah, that's a great question. So one of the, I would say, drawbacks of my work is I'm only addressing excretion as an input of nutrients. And so there's also digestion, feces, poop, of course, and then also dead crabs. And I think my leading theory right now as to why the green crabs might have these lower excretion rates. First of all, I should point out they're swimming crabs. They're not taxonomically similar to the rock crabs that we're looking at. And so different species just have different excretion rates. And so it's possible that these swimming crab species just have reduced excretion. Given their really high consumption rates, they're eating sometimes up to like 70% of their body weight a day in prey. There's a chance that that's just running through them and they're not actually digesting and metabolizing as much of that food. And so they're just eating it and it's not really getting processed and it's just shooting back out the other side. And so, yeah, between the poop and the potential piles of dead crabs, that would be a lot of food for bacteria that could potentially break down that matter and then recycle that back into ammonium. So that is another form of ammonium that could come back out. I'd be a little bit concerned about all that dead matter potentially causing like anoxic conditions, perhaps, but I haven't looked into, the decomposition of their bodies directly. That's a great question.
Hi, this is Jofina from Fisheries and Oceans. I was just curious about, kind of actually had a similar-ish question about the excretion rate. And you mentioned that the green crabs pee less because they're smaller, which makes sense. But have you ever compared like how much green crabs pee versus native crabs? if you have similar biomasses of both?
Oh yeah, so that is what this figure, I'm gonna whip through this, it's gonna be brutal. That is what we did in this figure. So the excretion rate is per gram of dry tissue. So come on. So they're peeing less, not only less per crab, but yeah, less per gram. I'm not sure that we're gonna get there. I'm gonna keep trying. And given like crab size relationships, a green crab that's the same exact mass of dry tissue mass as a native crab will actually have a slightly larger carapace size because they are just thinner shelled and we're so close. Maybe this isn't worth it. So what happens when you animate everything in. Okay, yeah, so for this figure, yeah, the log dry tissue mass, that is like per gram of dry crab, essentially. So both per crab and also per gram, they're lower excretors.
Hi, Christina with Okanagan Basin Water Board. Thanks for the talk. I have a bit of a different question that's been rolling around in my head, and I wondered if anybody is looking at sourcing invasives for food for humans. And so I just find that often invasives such as knotweed have really high concentrations of resveratrol. So that's a medicinal quality. And just thinking about these crabs and if there's any value.
Yeah, that's a wonderful question. So I've personally never eaten green crab, but I've spent a lot of time with someone who has, and they have this kind of silly story about a whole class of students trying to harvest the meat from these green crabs and having like hundreds of crabs and spending hours. And at the end of that experience had like a little bowl of like claw meat. And so there's not a lot of kind of tasty tissue, I guess, in them. You could absolutely eat them. And Dr. Brett Howard at the Department of Fisheries and Oceans is definitely someone to chat with. She gave a wonderful keynote at a conference a couple of years ago. And so I'm going to try to butcher what she said in response to a similar question, which is that one of the issues is that if DFO officially has a fishery for green crabs, it's in their mandate that all their fisheries are sustainable. And so that's a bit of a trick where if they open up a fishery for green crabs, then they actually have to make sure that there's still going to be green crabs, which is opposite to the goal of eradication. There's also some concerns around incentivizing catch of green crabs that sets up an unfortunate incentive for people to maybe move them around more and means people will actually want to have green crabs and access to green crabs so they can catch them. There are a lot of societies like the Coastal Restoration Society pulling like thousands, like tens and hundreds of thousands of crabs out of the ocean. And I think there's been conversations around using them for fertilizer. There's a kind of a funny whiskey that you can buy that's made with green crabs. And so I think there are pretty innovative solutions people are trying to come up with to use this biomass, because right now I think there's a lot of organizations with freezers and freezers full of green crabs. And if we're pulling out this biomass, yeah, it'd be wonderful. We could eat them or make whiskey out of them or fertilize our gardens with them. But yeah, I think it's something that at least Brad at DFO is thinking about. But yeah, it sounds like it might be a bit tricky.
Yeah, we tried to get that whiskey, I think, for the forum. But I think it's pretty tough to get actually on the East Coast. Yeah. Yeah.
You've been trying, right? Yeah. Okay. Just gonna whip right back through all this.
We can do it.
Okay.
All right.
Oh yeah, you got it. Thank you so much.
Give them a warm round of applause. And I think, correct me if I'm wrong, some researchers on the East Coast, I think, have devised a way to make bioplastic from their shells. I think I heard something about that. All righty. Okay, we're staying on theme with your European green crab. Our next speaker is Milène Wiebe, who is a MSc student from our keynote speaker yesterday, Dr. Green from her lab at the University of Alberta, looking at the invasion ecology and adaptive management strategies concerning green crabs off the coast of BC. Yeah, there you go. Welcome.
Thank you. Little did you know that there's a pop quiz after this on green crabs, so take notes. But yeah, so I'm gonna, there's been a lot of discussion about green crabs, so I'm gonna kind of go quickly through my introduction, where am I pointing this? Awesome. As we work with invasive species, we know that there's a lot of impacts, not only to ecosystems, but also to human populations. So due to these impacts, often a question is, well, how do we manage them? How do we mitigate these impacts? Oftentimes, it really depends on the geographic extent or the stage of invasion. We might have seen this slide yesterday from Stephanie, where if your boxes and the circles are your invasive populations, eventually you're going to get to a point where you don't have enough resources to deal with these invasive species. So this kind of leads to the question regarding, well, how do you optimize this management? It has two questions. So what control techniques and tools maximize invasive species removal, and what level of harvest is required to alleviate these impacts? So I'm going to start off with the first question. So control techniques and tools. Often when you're trying to optimize your strategies, it can vary depending on the invasive species that you're working with, but often it comes down to your approach. So with invasive plants, are you mowing them or are you hand pulling them? Obviously, that's a simplification. What tools are you using? So with trapping, is there a better trap or is there a worse trap? And lastly, timing. So is it better to deal with invasives in the spring, maybe before they start pollinating, or in the autumn when they start dying back? So for looking at optimizing strategies, I'm using green crab as a case study. You've heard a little bit about them today as well. Globally invasive, native to Europe and Northwest Africa, and one of the 100 worst invasive species. Within BC, they've arrived, or they're thought to arrive during the 1997-1998 El Nino, with adults being found in Victoria and Barkley Sound in 1999, and they have slowly moved their way north up to Prince Rupert and Haida in 2020 and 2021. Again, quick look at impacts. Obviously, altering habitats, specifically eelgrass, important fish rearing and spawning habitats. And of course, the consumption and just competition of native species, some of which are important commercially, other ones are quite important for coastal communities, not only for sustenance harvest, but for cultural reasons as well. Often with green crabs, we found that the best management is to go through trapping, with studies actually showing that with consistent trapping over time in some areas, you'll actually see a reduction in the number of green crabs that you've trapped. One issue is that often with these trapping programs, guidance is often being done without consultation of these impact communities, and certain guidelines tend to be less optimal for other conservation goals. So if we're looking at the guidance regarding trap type, a lot of areas are using these Fukui traps. They're collapsible, so they're easy to carry around, but they're rectangular with ramps climbing up into the cage, so green crabs actually have to squeeze through a little narrow opening. They work pretty well, but unfortunately, they are often associated with unintended mammalian mortalities, often seeing otters or mink when you're pulling up these traps, which is quite unfortunate. Same thing happens when we're looking at bait type. Often guidance is to use herring within your traps, and herring is now becoming quite pricey, which is if you're trying to have a sustainable trapping program, you want to use something that's less expensive, but additionally, there are concerns regarding herring stocks. And so to use them as bait to get rid of an invasive species is counterintuitive to your other conservation goals. So, in addition to this guidance, often when we're looking at the when we're looking at trap types, bait types, or other trapping variables, they're done singularly. So often a study will look at just trap types, just bait type, or other variables such as time or depth. And unfortunately, we're not really looking at the interactions that are occurring when we're testing out all of these variables. So it is in collaboration with the Syilx First Nation on the Central Coast that we're trying to address these issues. So one, are there alternatives to these current guidelines? And also, what are these interactions? How can we perfect these trapping programs? So we approached this in a two-part project. First, looking at alternative trap types. So the nation actually created or adapted pot traps to create this custom trap type in an attempt to avoid mammalian mortalities. And we've compared them against the standard Fukui as well as G-minnow traps, which are typically used to monitor juvenile green crab populations. Once we figure out which trap type is optimal, we then had conversations regarding alternative bait types. So again, as a reminder, herring tends to be the current guidance, but within the territory, they've historically hunted sea lions for sustenance, and they thought that maybe using the oily blubber could mimic the oiliness of the herring. So we attempted to use sea lion, and of course we needed to try green crab because it's a free bait that it's kind of a cyclical, where if you're trapping them, you can just crush them, put them back in the trap, and use them again. So to approach this study, again, done in the Syilx Nation, highlighted in red, and the nation identified a specific area that is quite important for commercial shellfish harvesting, highlighted in green, and trapping occurred over a suite of sites here. So Bar, Beach, the Cabin, and Canute. Quickly going over methods, we had alternating trap types deployed across a range of depths, and we use relative depth as kind of the average depth at deployment on a specific day. We did a lot of analyses regarding our variables. We didn't do anything with non-mammalian bycatch in the first phase, just because the sample size was too small. But but I do want to note that there was one mammalian mortality of an otter in a Fukui trap. So for this conference, for this first part, I'm going to focus on CPUE, which catch per unit effort. Here I'm defining as number of crabs per trap per day, and it's a function of trap type and relative depth. I created an incident rate ratio where the reference line is our standard Fukui trap. If we compare it against the custom trap, we see that the custom trap is outperforming by having a CPUE that is about 5.5 times larger than our Fukui traps, which is a good sign. When we look at minnow traps, they're not performing as well. They caught about a 1/10 of the CPUE that our reference Fukui did. But that makes sense because if they're targeting juveniles, you're not going to get a bunch of green crab kind of shoved into one trap. And then relative depth was not a significant factor within CPUE. So now that we know that our custom trap was the best trap type, we applied it to the second phase, where this is our initial layout for traps. We replaced all of the minnow traps with these custom traps, and then we tested out our 3 bait types. So a reminder, we had herring, sea lion, and green crab, in, again, alternating fashion. Here, I was also able to model a mammalian or non-mammalian bycatch, and we only had, again, one mammalian mortality, which was a mink in a Fukui trap once again. I'm only going to speak on CPUE and the number of non-mammalian bycatch, where catch per unit effort is defined as an interaction between trap type and bait type and relative depth, and non-mammalian bycatch is trap type with bait type, and then relative depth and length of deployment. When we do, again, our lovely incident rate ratio graph, with Fukui being our first reference point, Fukui traps still outperform, but only about 2 times. So they're still doing a lot better, just not to that 5.5 times larger. When we're looking at our reference for bait type, comparing against herring, we see that sea lion actually is only catching about half of the CPUE, and green crab again is 1/10. An interesting aspect, though, is when we're looking at the interaction where we have our alternative bait types in our custom traps compared to the herring within a Fukui trap, there is no significant difference. So if you still want to keep your numbers up, but you don't want to be using herring, these are some good alternative bait types. And lastly, again, looking at relative depths, we saw that with an increase in depth, you're seeing a decrease in the catch per unit effort. So just like Em said, they are preferring some of the more shallow areas. When we're looking at bycatch, an interesting thing, I'm just going to speed through here, is that we didn't really see a significant effect of either trap type on its own, bait on its own, or its interaction. We did see that the relative depth at deployment, the deeper you go, the more non-mammalian species or non-mammalian bycatch that you're catching, which again makes sense if the green crabs are pushing them out. So going back to our two questions regarding optimizing management, techniques and tools for green crabs, your best bet would be to use, if you want to optimize it, is to use a custom trap baited with herring, deploying in mid to shallow depths, if you really want to optimize it. So using this information, we're starting to ask the question about what level of harvest is required to alleviate these impacts. So again, some of this might sound a little familiar from Stephanie yesterday, but if we're thinking about optimizing targets, and we have a graph with invader density on the x-axis, on the left, you have your ecological benefit per additional unit of suppression, and it decreases as invader density goes up. On the right, if we have effort per unit suppression, you see this nice little peak, and it decreases again as invader density goes up. So we have this sweet spot right in the middle, where you have an efficient allocation of resources within this red zone, and either inefficient or insufficient effort being given. If we apply this to the green crab relationship with shellfish, again, it's a non-linear relationship that you're seeing. And based off of the work done by Ted Groshalvs in California, the relationship here is that you would expect to see your best, basically your best bang for your buck between 10 to 20 catch per unit effort or crabs per trap per day. Below that, it's inefficient, and above it is insufficient. But this work is done in California, and so we really wanted to, and based off of the work that they've done, theoretical, we wanted to test it within the field, asking, does this relationship hold true when we're doing it in a different area? So we approached this by using an exclusion and inclusion cages, a predator exclusion experiment. We chose this experimental approach over observational just due to you have a lot more control over your starting density of juvenile clams. Plus, it can happen in a lot shorter time frame. To conduct this, we chose 3 sites, 3 beautiful beaches. I wish this is what our beaches looked like. It was not. Across these 3 sites, we had 2 sites that had really high numbers of green crab and 1 that had, as of yet, no sightings. This is also another 2-phase project where we did a lot of sampling before we did any work, and then we trapped intensively for about 2.5 months to see if we could decrease the CPUE. So a quick methodology, after I'm ignoring all the digging that we've done, but we seeded each cage with juvenile clams, let them settle for 24 hours, opened the cage and let them sit for 24 hours for predators like green crab to come in. We sampled a quarter of the cage at 24 hours, 3 days, and 7 days. Again, after this first phase, intense trapping using the custom traps were done for 2 months, and we pulled up a lot of crabs. After this point, we had the sequel of sampling, seeding, and sieving throughout this time period. Unfortunately, since I'm not only interested in the number of surviving juveniles, I'm also measuring, is there a size that is there a difference in the size of surviving clams? I'm still working in the lab trying to measure all of these clams and identify them. So I'm really excited to find or to share these results once I get them. I do want to acknowledge the Syilx Nation for the invitation to work on this project, as well as the knowledge holders for their insight and everyone who worked out in the rain doing all this trapping, especially in the middle of January. Thank you. And if there's any questions, I'm happy to answer them.
Hi, thanks. Em from Simon Fraser University. Awesome talk. Very cool to hear more about crab stuff. I'm just curious about the field experiment. So can you maybe just explain your 3 treatments again? And then maybe just specifically like how, I guess, would those results help determine the like 10 to 20 CPUE part?
Yeah, so our goal is to get down to 10 to 20 CPUE. Monitoring prior to this experiment was conducted to see what is the baseline CPUE at these sites. We have a site with no green crab to kind of monitor what would the natural predation in an uninvaded site be as kind of our baseline comparison. Then we wanted to see, well, if there could be other environmental conditions that cause change in population, such as temperature. And so we wanted to have kind of a control site to see, well, is CPUE changing without intense trapping, and how is that impacting survival? And then we're just trying to decrease CPUE as much as we can in this first site by trapping. Yeah, and so while the goal is 10 to 20 crabs per trap per day, we acknowledge that we might not get down to that point, and more intensive trapping might need to be done. Yeah, thank you.
Hello. I was just wondering about the graph where you were talking about like the inefficient use of energy versus the insufficient use of energy. I was wondering what the difference between those two were, and if insufficient is just another way of like saying like you don't need to keep trying to catch them because they're eradicated or something like that.
That is a really good question. I kind of did speed over that. So this one or the one before? This one? Perfect. So essentially what this graph is trying to convey is if you have a green crab catch per unit effort and your trapping isn't decreasing that CPUE enough, you're not really going to see a major return in the shellfish community. If we're looking, so your effort is insufficient at that point. If you are trapping below 10 green crabs per trap per day, you can be expending a lot more energy, but you're not seeing that much of an increase in your returns. So let's say like if you have 1 boat out versus 2 boats out, if essentially the result is going to be the same between that 1 and 2 boats, maybe that resource could be better spent in another area or on a different project. Yeah, good question.
Thank you so much to the speakers in our session here. We are going to have our break now, but before we do that, I just want to acknowledge and give a special thanks to Tech for sponsoring today's coffee and networking break. So yeah, very grateful for their support. So yeah, we'll be back here at approximately 10, what did we say? 10:45. So yeah, please give one more round of applause to Milène. We had M and we had Sandra. Sorry. And oh, sorry.
I forgot to mention that I have a funded PhD position in the synthesis research medicine.
Cool.
Spread the word.
you want to send an email to Nick or whatever, we'll circulate that across our network. So I was looking for a post-op.
Alrighty, thanks very much. See y'all back here shortly.
Thank you. All right, everyone, it is time to get started on our lightning talks. Welcome back. I hope you enjoyed your snacks. I saw lots of fresh fruit, which I was stoked about. We've got a little bit of a different session next, and I think this is going to be really fun. There are other names for these types of talks, but we call them lightning talks. Each presenter will have about 5 minutes. They're really short talks, just focusing on the key highlights. So that brevity is really important. There's some diverse topics that we're going to cover here, and hold on tight. Let's roll. I'm going to first introduce to you Dr. Jen Barron. Jen is a postdoc researcher, and congrats on being an incoming assistant professor in fire ecology at the Center for Wildfire Coexistence at the University of British Columbia in Okanagan. Woo! I feel like that needs a little cheer. Jen completed her PhD at the UBC Faculty of Forestry, where she focused on understanding and managing altered fire regimes in southeastern BC. Her research aims to understand how the ongoing legacies of colonial land management systems have altered fire regimes and how the strategic applications of treatments and management can restore fire-adapted communities, ecosystems, and landscapes. Please welcome Dr. Barron to the podium and her presentation, Balancing Wildfire Resilience, Wildlife Habitat, and Invasive Plant Management Through Ecosystem Restoration in Montane Forests.
Thank you. Yeah, thanks for having me. I'll try not to preamble too much because I only have 5 minutes, but very happy to be here. I got my start in ecology working in invasive species dynamics in Southern Ontario. And so I'm quite excited through my research program at UBC Okanagan to begin working in these systems again, but in a different way. Okay. So I work largely in a portion of the province where we have dry forest systems very similar to the Okanagan, but in the Rocky Mountain Trench and across BC, we know that historically Indigenous fire stewardship maintained these ecosystems in their open condition in the past. So this is an image of Tobacco Plains, which is right by the corner of the province. The Alberta border meets the US border. And you can see in the background, these are very open forest conditions. This is a fire scar record from near that ecosystem. It's the record for the number of fire scars in a ponderosa pine in North America. So this tree survived 52 fires during its lifetime, which is only possible due to the active role of fire that was created and maintained by Indigenous people. In particular, in this location, our understanding is that this burning was specifically for the cultivation of tobacco as a medicine plant. And this is a map that we developed during my PhD work that represents how departed these ecosystems are from their historical fire regimes. So it's a ratio of how often these ecosystems used to burn in the past relative to how frequently they burn today. The larger the number, the more fires are missing from that ecosystem. So particularly in our low elevation valley bottoms, we know that these ecosystems used to burn the most frequently in the past, as frequently as every 2 years at the site, and they haven't burned in over 100 years. So the low elevation is missing at least 10 fires, which creates a big challenge for wildfire management and also for ecosystem restoration. And this is specifically the region near Tobacco Plains where we're working on this study. So there's a couple factors that come together at this site. Fire exclusion has degraded these ecosystems. And so there is an overabundance of trees, which contributes to elevated wildfire risk as a consequence of our very successful approach to putting out all fires over the past 100 years. These ecosystems are also essential habitat for wildlife species, including mule deer, elk, and bighorn sheep. And in particular, for bighorn sheep, which face multiple threats from different sources, including disease, these dense forests obscure their access to escape terrain from predators, and they compromise the quality and abundance of forage due to canopy closure and the overstory. However, many of the treatments that would potentially improve these habitats also create concern around their introduction of invasive plant species. So we know from a fuels and fire behavior perspective that there's a number of evidence-based measures that allow us to mitigate potential fire behavior by removing fuels from these ecosystems. So these include things like forest thinning operations to remove overstory fuels, cultural and prescribed fire to reduce surface fuels, and manage wildfire, which occurs when a natural ignition ignites on the landscape and we allow it to burn up to designated perimeters for ecological values. However, all of these in their own way can also create opportunities for invasive plant species to proliferate, which complicates management decisions. In particular, at these sites near Tobacco Plains and in the Galtons Range, we're interested in mitigating wildfire risk while managing for invasive plants and habitat. So in particular, we're quite concerned about the presence of cheatgrass, which is highly flammable and creates continuous fuels, and is often a strong competitor in these post-fire environments. And we know from research in the Western US that in particular, these invasive grasses do very well with fire, where fire continues to facilitate invasive grass cycles at the detriment of native plant species. In addition, we also have concern over the abundance of St. John's wort in these ecosystems, which has a very deep and extensive root system and can have its propagation stimulated by fire and other disturbances. So these are two species that we're particularly interested in managing for. And so what we're trying to do in this project is thread the needle between reducing wildfire risk, improving wildlife habitat while mitigating invasive plant species, recognizing that the current condition of these forests is highly undesirable and it's not meeting values for any of these objectives. So we're trying a variety of logging and mechanical thinning treatments with and without prescribed fire, manage wildfire, as well as looking at herbicide and seeding and other invasive management strategies in combination. And then we're monitoring and measuring the effects on fuels and fuel treatment efficacy at reducing wildfire risk. We're looking at vegetation community composition and invasive and cultural plant abundance post-treatment. We're also doing forage and ungulate dietary analysis. And then we also have a large network of telemetry data from collared animals as well as camera traps across this landscape. So this project just started. It'll be ongoing over the next couple of years with fieldwork starting this summer. And I'm really looking forward to trying to address some of these challenges and concerns that create barriers to us implementing restoration work in this part of the province through this study. Thanks.
Questions? We have time for one question for each presenter. All right. Well, Jen, setting
a different agricultural setting. This is a native forest in the mountains of the Okanagan Valley, littered with black huckleberry plants on the forest floor. This plant has less economic value, and more cultural value to the Syilx people of the Okanagan, as well as Indigenous people across all of Western North America, the native range of this plant. Both of these plants share a common enemy. This is spotted wing Drosophila, an invasive fruit fly introduced from Southeastern Asia, with a adaptation highlighted in red here that allows it to slice into the flesh of ripe and ripening fruit and lay their eggs within. This creates a pathway for pathogen entry, not to mention the developing larvae within the fruit. Typically, the fruit will look okay for a short period of time, after which they will very rapidly decay. In order to investigate the interactions between these species, we have placed a number of sites across an elevational gradient within the Okanagan Valley and are collecting a number of data types. We've installed data loggers to monitor temperature and humidity within each site. We've installed fly traps to monitor spotted wing populations. We're monitoring the huckleberry plants themselves, and we are harvesting and rearing huckleberry as well as other soft-skinned fruits to inspect host use by spotted wing. The primary variable that we'll be working with in this modeling system is called the degree day. In its simplest form, this is an accumulation of heat energy. The plot on the top I've put together to try and illustrate what this can look like. This is the changing temperature throughout one day, illustrated by the orange line. The gray box is a temperature range bounded by the developmental thresholds required by the organism, and I've highlighted in red the accumulation of that heat energy in one day, and that's the degree days from one day. This is summed with the following day and every day throughout the rest of the growing season to produce the total length of the growing season measured as a standardized degree day metric. On the bottom plot, I've included the total number of degree days at each of my sites, plotted against their respective elevation to illustrate how strong this relationship really is and why we've used elevation in our experimental design as a proxy to measure different degree day development thresholds. We'll be using our degree day values in order to model the phenology or the life cycle progression of these two organisms. An example of some of the phenological milestones that we're interested in looking into are the beginning of the huckleberry fruiting season, the peak of huckleberry fruit abundance, and the duration of this season. On my last slide, I've just included a representation of how we might use one of these models, and then some of the questions we hope to answer through this modeling system. First of all, we'll take our predictor variables and input them into the model, which can then produce site-specific predictions for huckleberry phenological milestones. That's one of the models that we are going to produce and work with in order to answer some of these questions, such as when will this site in particular produce the most fruit? Is the timing of huckleberry fruit ripening going to coincide with the spotted wing population abundance? We heard a little bit about phenological mismatch and incompatibility earlier. And is this elevation a suitable location for the future propagation and cultivation of huckleberry sites? And through this, we hope to improve food sovereignty in the Indigenous people of the Okanagan. Thank you very much. I'll take any questions you have. With an extra 30 seconds. We need to run a microphone. Oh yeah, thank you.
Hello, this is Brennan from SFU, and I was wondering, so you're looking at this question because you're interested in the value of these fruits for people, right? But I just was also curious if I was curious how much faster they rot, like the fruits rot with the fly, and if that might also impact wildlife use.
Certainly, yeah, that is quite a lot faster. I don't have a metric for that offhand, but it could absolutely have an impact on bears in particular use huckleberry as a big food source. Thank you for your question.
All right. Awesome. Thank you.
1, 2. Test 1, 2.
Okay. Okay. Thank you for the introduction. Yeah, my name is Owen, and today I'd like to share with you some research that I conducted over a summer USRA. And this may be a bit silly, but I really need to sell you guys on this part. So imagine you're out on a beach. It's a nice sunny day with a cool breeze. There's kites in the sky. You think to yourself, it's the perfect summer day. Friends and family are with you. And you're going out, you're going to take your first step out on the beach, expecting nice, warm sand. I think folks from Kelowna can understand, right? But instead, you get a sharp pain in your foot. And that's when you look down and you realize that the ground is covered in thousands, if not millions, of these cone-shaped snails. And this is the invasive Japanese mud snail, or Basilaria atramentaria. They've been an invader on the West Coast since the 1900s and can reach absolutely crazy densities and dominate beaches. Many researchers have looked at their impacts on other species, but no one has looked at how this mud snail affects sediment. So that's what we set out to do. Before we started, we knew that Basilaria could affect sediment directly through bioturbation, like these snail trails here, or by loosening up sediment in search of food, or by burrowing into the sand to avoid drying out. And we also knew that they could indirectly affect sediment through what they eat. Sorry, I should be talking here. Since diatoms, their food produces sticky mucus that binds sediment together. With this background knowledge and our predictions, we set out, and in the summer of 2024, we conducted a 10-week-long caging experiment in Blackie Spit, Boundary Bay. At Blackie Spit, we set up 40 plots of differing snail density, mid being our ambient, high being double the ambient, and low being half of it. We also had in place 2 controls, a no-cage control to test the effect of the cage itself, and a standard control plot, which is just the cage with 0 snails. And throughout the experiment, we maintained the preset densities. In each cage, we measured chlorophyll concentration, which we're using as a proxy for diatom abundance using a chlorophyllometer. We measured sediment penetrability using a dart by seeing how soft or how hard the ground was, and erosion by looking at the change in height of these tubes over time. So after the 10 weeks, what did we find? And looking at chlorophyll concentration, we see on the x-axis time, and on the y-axis chlorophyll concentration in CFR. The dots represent raw data. We have a fitted line in the gray, and we have a gray confidence interval. And in our control plot, we can see that chlorophyll concentration increases over the duration of the experiment. Now, for all the treatments, our densities are grouped separately from low on the left to high on the right, so we can easily tell the difference between them. When compared to the control, we can see that increasing the number of snails does not have an effect on chlorophyll concentration. And we see a similar trend when we look at sediment penetrability, or how soft or hard the sediment is. On our y-axis, we have our measurement of sediment penetrability. Higher values indicate softer sediment, lower values indicate harder sediment. And again, in our control, sediment is getting softer, but when we add snails to it, that doesn't really change it. Erosion is a bit harder to understand. All you should know is that if the line is above 0, we have sediment accretion, which is more sediment being added. And if it's below, we have sediment deposition, which is sediment being taken away. And we can see, once again, when compared to the control, snails aren't changing the rate of accretion. So what's happening then? We didn't see an expected effect of density on our variables, but still we did see an increase in chlorophyll concentration. The sediments in our cages got softer, and we had sediment buildup in our cages. So why is this happening? And the answer is pretty clearly the cage. Looking at our control, which is our cages with no snails, and our medium density, which are the cages with the ambient density of snails, we see that having snails doesn't affect chlorophyll concentration. But when we compare medium density cage snails with our no cage plots, which have the ambient density of snails, so those two are comparable, we can see a large effect of cage, as within them, chlorophyll concentration increases, but outside, there's really no change at all. And we see a similar pattern with sediment penetrability. Sediment is getting softer inside our cages, snails aren't changing that, but outside, there's no change at all. And again, we see that with erosion. Inside the cages, we're seeing accretion, adding snails to them doesn't change that, and outside the cage, we see deposition actively happening. So what does that mean for the snails? Are they having no effect even when they can reach these absurdly high densities? Not necessarily. It could just be that we're looking at different variables and they're affecting different ones. And there's still 2 lab procedures we have yet to finish the procedure on. Those being diatom assemblages, like how many diatoms there are and what size they are, and also a direct measure of the sticky mucus. And it also could be that the cages just provide too much of an effect and overpower the effect of the snails. So with that, I'd like to conclude and just thank you for listening, and thank you to all my project partners for helping.
Great presentation, Owen. As an avid kayaker, you did not have to sell me on that first part about the challenges on a beach. My partner and I actually have a code for this. SMS means so many snails, not a good beach. Any questions for Owen? All right. Oh, we got one with Dr. Green.
Thanks, Owen. That was a great talk. Cage design, I'm sure you've thought about this a lot. How might you alter the experimental device?
I didn't mention this because I adapted this into a 5-minute talk, but we had a lot of intruders in our cages, whether that was other invasives because Boundary Bay is full of them, or shore crabs, which really just seemed to prefer the cages because I think they were protecting them from birds. So maybe make them deeper into the sediment or find a way to exclude stuff as much as I want to include the snails in them.
All right. Thanks again, Owen. Really appreciate you being here with us today to share about your work. All right, our final presenter of this session and of the day before we move into our workshop and of the entire conference. What an honor. So happy to welcome Andreas Mancera Barreto, a master's student here at UBCO and working with Agriculture and Agri-Food Canada in Summerland, another organization that we've heard about a lot here today. So thanks for all of your leadership here. Since 2022, Andreas has been merging his interest in entomology, insect-plant interactions, and ecosystem conservation to study noxious invasive weeds in BC. As a graduate student, he's had the chance to work in one of the oldest and most successful weed biocontrol systems in Canada, helping solve questions to help ensure biological control continues to be an effective tool for the management and health of BC rangeland ecosystems. Andreas's presentation is eligible for CECs if you're here with the BCLNA. Andreas, really excited to hear about identifying geoclimatic factors and biological interactions influencing St. John's wort biological control.
Thank you all for having me, and thank you for the introduction. I'm here to talk about St. John's wort. But before I do that, I will piggyback on a bunch of previous presentations, and I will assume that everyone knows why invasives are bad and why we want to control them. So what is biocontrol? We want to introduce a natural specialized enemy that is really good at dealing with whatever invasive we are dealing with, because there's a lot of food available for this newly introduced agent. They rapidly shoot in population, and eventually they have this dynamic interaction between invasive and control, leading to an overall decrease over time to a threshold that we set up on the ecosystem. Oh, my slide. So what does this has to do with St. John's wort? St. John's wort is Canada's oldest insect plant biocontrol system. It is also a flagship system. It is extremely successful, and it is very important because St. John's wort is a toxic plant that has a phytotoxin that after ingested makes the animals sick. So ranchers hate it, the cattle hates it, it dominates grasslands where we have our cattle, so it is important to control it. It overall reduces forage quality. So after pressure, what we did is we introduced several species, of which 5 established, and they are fantastic dealing with this plant. They have behavioral and morphological capacities that allows them to deal with the toxin, so they don't worry about that. They attack several parts of the plant, so they are doing an amazing job, which we can see right here in these pictures. 20 years of difference, and it's just basically gone. Extremely successful, and we do have data from back then that backs the success. However, why do we have renewed interest on the St. John's wort? Well, we have data gaps post-release. Long-term monitoring wasn't done, and although we do have some data points, we need more to be able to understand how the system is changing, especially thanks to environmental and climatic changes. As we all know, climate change is happening. It is impacting everything everywhere. So it is important to know if our biocontrol agents are being stressed due to new conditions. So we need to reevaluate the strength of the system, and hopefully we are able to understand the system better as it is changing. So to understand that, I am looking to a bunch of different variables out in the field, such as growing season length, which is an accumulation of growing degree days when Nicholas talked about, so I'm going to piggyback on that. Elevation, because it is important to know the geography of a site, humidity, geography, abundance, and herbivory rates. So I am recording the local climate and the site geography of all of my sites, and a bunch of biological interactions with our insects and plants. So plant growth, plant phenology, insect phenology, the abundance of both of them. Abundance on St. John's wort is weird because it's a clonal plant. So what is one individual gets funky? So we measure coverage and herbivory rates, so on and so forth. We are measuring that across 17 sites on the Okanagan, ranging in about 1,000 meters in elevation. And what we are expecting to see is that due to new concerns of climate and new reports of the weed coming back, is that if biocontrol is lacking, something like the green line will happen, where we see a lot of abundance at low elevations where the plant is really successful, and then it decreases with elevation. If there is some partial biocontrol, then we expect the pink line, where we have some strong control at the bottom, but then it decreases. And then if we are being successful, we expect the purple line. Although we are just starting to analyze our data, we are seeing some nice results like this. We are seeing less plants lower down where we were expecting to see more. Overall, the coverage is really low for an invasive. So although all of this is preliminary, all of this is just one season, it's indicating that possibly biocontrol is still being quite effective on this plant. Still, lots of variables yet to model. We are just getting started on this, but so far, so good, I think, I hope. With all of that, thank you very much for having me.
Any questions for Andreas? Last chance for questions in this part of the conference. I know you gave an amazing poster session the other day too, so maybe some of those questions got explored then. I know I certainly had a really great conversation with you. So, all right, Andreas, one more round of applause. Thanks for doing such a great job of finishing off our the presentation section of our conference, and thanks to all of you for being here both in person and online. Okay, so we are going to take a short break and come back at 11:25 to start the workshop section here, but I just have a couple of quick housekeeping items.
Do people want a break, or do you just want to go straight through? Just a question.
I think we might need some break time for online. Yeah. Okay. So we are going to take a 10-minute break. But before we go there, I would love to just have everyone join together for a round of applause for all of our incredible presenters. So many great things learned. And let's get some cheers in there too. Woo! 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 Okay, awesome. I'm going to ask for two more just like that. I want to shout out the amazing volunteers that are here. We've got people timekeeping, answering questions online, running the mic. I think that's been incredible, helping organize and set up. So thank you so much to our amazing volunteers. We super appreciate you. All right. And once again, our sponsors, we couldn't do it without you. Thanks for being here. Thanks for all of the ways that you're helping us connect and share our learnings. Woo! All right, let's break, come back here at 11:30 and we'll get rolling on our workshop, and we're still going to be out of here on time as planned. So yeah, grab something to drink, take a bio break, and we'll see you shortly.